Alright, welcome to part 4 of this lecture, and here we're going to consider secondary, tertiary, and quaternary levels of protein structure which are necessary to result in a final functional protein. So we're going to talk about how peptides form proteins. So first let's define a protein. A protein formally and correctly is the complete, often functional, form of the molecule. Functional proteins have specific three-dimensional structures and in order to do their work, and functional proteins include one or sometimes more peptides. So let's think about protein structure again. We talked about primary structure, which is the identity, sequence, and length of a peptide within a protein. We discussed that in the previous section of this lecture. Now we're going to think about secondary, tertiary, and quaternary. I want to note that not all proteins achieve quaternary structure. Some, once they achieve tertiary structure, are fully functional. So first we'll talk about secondary structure. This is the layer of structure in protein that is created largely by hydrogen bonding, which is what I mean by H bonding, between components of the peptide backbone. So shown here in green is the peptide backbone, and we discussed that structure and the terminology that's important for you to know there. And if you look at each amino acid residue, you can see the central carbon. Here's the central carbon here, and you can see what used to be a carboxyl group. There's a peptide bond. Here is the nitrogen that was in the amino group for this amino acid residue, and so on. So the hydrogen bonding that's important in secondary structure is connecting portions of the peptide backbone other than the R group. So the R groups are shown here, 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 and so on. The hydrogen bonding is happening between oxygens associated with the carboxyl groups, hydrogens associated with the nitrogen group, and sometimes it can happen with hydrogens that are associated with that are simply covalently bonded to the central carbon. So this is an internal kind of bonding. Remember, these aren't flat static structures, they're flexible and they twist. So this hydrogen and this oxygen would have to come together in space in order for this hydrogen bond to form, and that can happen due to the flexibility of the protein or the peptide backbone. So here are some examples of the secondary structures that are very common in nature. They have very distinctive shapes. So a very common kind of shape is the alpha helix, shown here, and the beta pleated sheet, shown here. And you can see the internal hydrogen bonding that is causing these the formation of these structures to occur. So the shapes that form as a result of secondary structure, as a result of this internal hydrogen bonding, depend on the primary structure of the peptide, the identity and sequence of the amino acids in the peptide. It's important because it controls, the R groups control how the protein flexes in a cell, because those R groups will interact differently with the water always found in cells, and that will affect how the peptide flops around or twists, and that will affect whether hydrogen bonding can occur or not, and therefore control the formation of secondary protein structures. There are other types of secondary structures than the ones shown, but these are the most common and the ones you should be familiar with. So tertiary structure is the third level of protein structure. It's due to additional folding that is imposed by chemical bonding that occurs between R groups and also between many areas on the peptide itself, R groups and other functional groups on amino acid residues. Many different kinds of chemical bonding influence this kind of structure, this layer or level of structure. These are hydrogen, covalent, and ionic bonding. We already defined and discussed those in the previous lecture, and also hydrophobic interactions and van der Waals forces, which we haven't talked about yet. Hydrophobic interactions occur when hydrophobic non-polar R groups, and note that R groups are also called side chains, are in the water solution found in all cells, and the water interacts strongly with itself and with polar side chains, causing the nonpolar R groups to be pushed together into globs. Essentially, the nonpolar side chains are surrounded by so much other material that is all hydrogen bonded together that they are forced to clump together, and that's what we mean by hydrophobic interactions. Van der Waals forces are a little bit different. They're a type of electrical interaction that occurs within nonpolar molecules or portions of molecules as a result of the continual movement of electrons within any atom or molecule. So this figure, which is figure 3.11 of your text, whoops, need to move there. This figure, which is figure 3.11 of your text, depicts the many different types of chemical bonding that are important in tertiary structure. So notice that the ribbon is a peptide, or actually more correctly, a term called polypeptide applies, which is a longer peptide. You can see your text for the formal definition. So on one end of that green ribbon there's an N terminus, say over here. On the other there's a C terminus, say over here. And we've already had some level of secondary structure imposed on the molecule. Now we have tertiary structure, and the tertiary structure is happening as a result of many different kinds of chemical bonding. So we see hydrogen bonding over here between a side chain or R group. Remember, side chain is the same as an R group. Okay. We have hydrogen bonding occurring over here between two side chains or R groups, okay? And we have hydrophobic interactions and van der Waals forces interacting here, ionic bonding over here. This is something called a disulfide bond, which forms, it's a very strong covalent bond that forms between two sulfur atoms from sulfur-rich side chains, such as, I think that could be methionine. You can check your text to be sure, check up on me. So these are all the different kinds of chemical bonding represented in this figure that impose this third level called tertiary structure onto a protein. Now once this happens, many proteins are in fact fully functional, and that is in fact the complete finished protein able to do its work in the cell. Tertiary structures are very diverse. Those are some representative ones shown there for a variety of proteins out there. The structure will be correlated to the function. In other words, there'll be a structure that suits the function of this particular cell. And you should remember also that each tertiary structure equals one polypeptide. There's only one chain in there. Okay. Now I said for many proteins tertiary structure is the final level of structure, but not for all. Some go on to what is called quaternary, or the fourth level of structure. So here are two proteins that have achieved quaternary structure, and they are only functional and therefore complete proteins when they are in this structure. So quaternary structure occurs when you have the binding of more than one, or the bonding of more than one polypeptide to form a functional protein. Only some proteins achieve quaternary structure. So on the left we have something called crow protein. It's referred to as a dimer because it contains two peptides, two polypeptides. Each of those polypeptides had gone through primary, secondary, and tertiary structure, this one and this one, and now the two are chemically bonded together. The nature of those chemical bonds can vary depending on the protein. Often they're just hydrogen bonds though. Here we have hemoglobin, probably one of the best studied proteins on Earth. It's a tetramer. It has 4 polypeptides associated with it. 2 alphas, which are identical, and they're shown that way as green, and 2 different types of betas. They're very, very similar, but they do differ slightly. So there are 4 polypeptides there. 2 are identical. The alphas are encoded by 1 gene, same gene, utilized to form both of those. And the beta 1 and the beta 2 each have their own gene. So one gene encodes that, and one gene encodes that. This would be in a normal human adult, for example. Therefore, there's a total of 1 plus 1 plus 1 equals 3 genes that are required to form the 3 polypeptides that are necessary to form that hemoglobin protein. And the Crow protein, they're both identical, so it's only the same gene. We'll get to talking more about that as we move onwards in the course and you learn about the genetic message and start to develop a more sophisticated understanding of what a gene is. Okay, so I'm going to finish this part of the lecture by just mentioning that it is essential that proteins fold correctly to achieve a very particular three-dimensional shape in order for them to have correct function. And in the last decade, maybe decade and a half, we've been learning a lot about some very tiny proteins that are called prions. And we're interested in prions because when they are folded incorrectly, this form over here, they cause a variety of very infectious, very dangerous diseases. They're not infectious in the sense that they could be transmitted in the regular way from person to person. Instead, you can actually obtain these infectious proteins when you eat contaminated meat. So humans, sheep, cows, goats, and some other animals can suffer from diseases. For example, spongiform encephalopathy, which is a degenerative brain condition. Now, all of us right now will have normal prion protein in our brain, and some studies are indicating that when it's normal, it plays a role in developing memory and in healthy learning. But something can trigger a change in the conformation or structure and an incorrect folding of the prion protein, resulting in this infectious form. And what's really neat is that when one of these infectious prion proteins encounters a normal prion protein, the infectious one will actually cause the normal one to change shape and become infectious. So if you eat some contaminated meat and you get these infectious prion proteins, even one or two of them into your body, they will make their way to your nervous tissue, into your brain, and then they will slowly proliferate, causing all the normal prion proteins to become diseased. It's a very scary kind of condition that we're really just starting to learn about. So the message here is that correct protein folding is necessary for correct function. And with that, I will conclude the 4th of 4 parts to Lecture 2 and end this lecture.